Well, welcome, Dr. Tommy Verbuchen. And I will stop sharing if you want to jump in with your slides. I was just focusing on that. I think that's your first figure that we've been thinking about. But we're really excited to have you. I'm really glad really glad you got your voice back and are here to talk to us today. As you know, we as a group are super interested in diversity and the outbred lines and some of the technical techniques you use are really at the center of that. So we're very excited to hear the specifics of what you have to share with us today. Thanks so much for coming.
Well, thanks for the invitation. Excited to be here. And as I told you, I've always watched several of these on YouTube, and so I really enjoy the seminar series in general. So I'm excited to share some of our work today. You guys can see my slides.
Yes, we can.
Great. Okay. And I think along the way, if you guys have questions, feel free to just jump in. I think it's easier to do it that way rather than waiting until the end.
Great.
So I'm Tommy Verbuchen. I'm an assistant member in the developmental biology program and the Center for Stem Cell Biology at Sloan Kettering in New York City. And I'm going to tell you about our work today, developing genetically diverse stem cell models from mouse outbred stocks and modeling brain development in those pluripotent stem cell lines. So I don't think I need to introduce this too extensively to you guys, but there's tremendous interest in understanding the nature of human complex diseases. And so over the last 15 to 20 years, extensive human genetic studies have been performed to understand the genetic variants within human populations that contribute to the risk of developing neurological diseases. So for example, here I'm showing a GWAS Manhattan plot from schizophrenia GWAS study from 310,000 individuals. And, the kind of take-home message from these studies is that all of these common disorders are incredibly genetically complex. They are massively polygenic, meaning that hundreds or thousands of common alleles and rare alleles within the population are contributing to the genetic component of risk of developing these diseases. And this is, maybe not unexpected for someone of my generation, but 20 or 30 years ago, I think the thought was that the genetic architecture of these diseases would be much more simple. And so, given this amazing genetic data, there's now a tremendous interest in understanding how these variants are actually modulating brain development and being able to model these biologies in pluripotent stem cells from humans. And so just to make things even more complicated, the majority of these common variants that modulate neurological disease risk are found in the non-coding genome. So they're not modulating protein function directly, but rather changing the timing or levels of expression of genes throughout the genome. And so, in a big picture sense, some of the big gaps in knowledge in the field are trying to understand how inter-individual genetic variation, especially cis-regulatory variants within natural populations, contribute to quantitative variation in brain structure and function. And in order to do this, we really have to understand how these variants are modulating gene regulation in the context of the gene regulatory networks that are controlling neural cell fate specification and differentiation. And then, through those gene regulatory changes, you get molecular and cellular changes, which sort of compound into this structural variation and ultimately functional variation in the brain. And so this is a very significant challenge because brain, as we all know, brain development is very dynamic and complex. And so here I'm showing you a picture, of course, of a human cerebral cortex, but this is actually the mouse progenitors, embryonic progenitors of the cerebral cortex, where you have dynamic changes in the behavior of these embryonic neural progenitors, where they, initially self-renew, then give rise to different sets of deep layer and upper layer neurons in the cortex, and then followed by oligodendrocytes and astroglia. So in order to understand how genetic variation contributes to phenotypic variation, we have to understand how genetic variation contributes to differences in gene regulation during these dynamic programs of development, which then ultimately lead to changes in the adult brain and its function. And so this is sort of conceptualized classically as kind of a genotype phenotype problem. So we don't want to understand how genotypes contribute ultimately to phenotypes, in this case in the brain. And so this has been conceptualized as kind of thinking about how one can have a space in genotype space, and then how that ultimately connects to an adult phenotype. And of course, in order, the genotype is manifests into differences, and builds the embryo through the process of development. And I think a critical point about modeling development is that it's actually a very critical advantage is that pluripotent stem cells enable longitudinal studies of embryonic development. So if you want to study a specific stage of mouse brain development, you ultimately have to sacrifice the mother and take the embryos out to analyze them. But that means that you can only look at one specific stage for any given set of embryos. And that makes it very difficult actually to perform large-scale genetic studies of developmental processes. But pluripotent stem cells, because we immortalize individual genotypes, it allows you to actually repeatedly sample development across a wide variety of stages, at least to the extent to which they can be recapitulated in vitro, of course. So when I started my lab at Sloan Kettering, we sort of had this goal in mind of developing a pluripotent stem cell platform that would allow us to map quantitative trait loci that impact brain development using genetically diverse outbred stock of mice called the diversity outbred mice. And so in the big picture sense, the diversity outbred mice, which I'll tell you about in much more detail later, we would like to have a panel of genetically diverse stem cell lines from this stock, ideally something in the ballpark of 200 to 400 individuals. And then using that panel, we could differentiate them in vitro into complex multicellular models of brain development, such as neural organoids. And then utilizing things like single cell RNA-seq and immunofluorescence, we can phenotype those organoids and ultimately identify quantitative trait loci that contribute to phenotypic variation in molecular and cellular features of those neural organoids. But there were some significant challenges when I started my lab that made this not a trivial undertaking. And so one was that the current or the available protocols at the time for directed differentiation of mouse pluripotent stem cells into neural cell types were very suboptimal compared to what was possible in human pluripotent stem cell field. And similarly, there was a limited availability of genetically diverse pluripotent stem cell lines that we could use for those studies. And so in a broad sense, an outline of my talk will be discussing how we've been addressing those two major challenges to develop this platform to be able to map, use sort of systems genetics approaches in this diversity outbred stock to map molecular and cellular phenotypic variation in brain development. So in the first half, I'm going to tell you about mostly work from a postdoc led by a postdoc, Danny Medina Cano in the lab, working together with Tarek and Veronica, who are graduate students. And this is a project that was co-led by Emily Wong, who's a computational biologist at the Victor Chang Cardiac Research Institute in Australia. And this work has been published. In case you're interested for additional details, information's at the bottom of the slide. So as many of you know, the original mouse the original brain organoids were actually made from mouse pluripotent stem cells and not human pluripotent stem cells by Yoshiki Sasai and colleagues in the early to mid 2000s. And so in these studies, Sasai and his colleagues showed that mouse embryonic stem cells can be aggregated and then patterned into several distinct types of brain tissue. When they tried to make cortical organoids, they had several issues. One was that they had trouble consistently specifying telencephalic identity in these mouse ESL-derived cortical organoids. And this was most of that work was done prior to the technical advance that adding Matrigel to the culture allows these organoids to form in the proper inside-out formation, with the basal side on the outside and the apical side on the inside. And so this was really pioneering work, and it really underpinned all of the subsequent advances in the human organoid field. But the actual mouse brain organoid protocols themselves were the degree of difficulty in getting them to work is quite high, and they have not really been utilized significantly by others in the field since then. And so most people in the brain organoid field have been making their organoids from human pluripotent stem cells. And so to make a long story short, one of the major differences in what Sasai was doing with mouse and what people in human were doing is that they're actually starting from a different pluripotent state. And so this terminology is really annoying, and I have to explain this over and over again. Unfortunately, this is the way people decided to do it. So mouse embryonic stem cells, when you say that, that refers actually to cells cultured in the naive pluripotent state. Whereas if human embryonic stem cells refers to cells cultured actually in the prime pluripotent state. And so in mouse, the naive pluripotent state is prior to implantation, the blastocyst stage, and then after implantation, the cells transition to this prime pluripotent state in the post-implantation epiblast, which in mouse are referred to as mouse epiblast stem cells. And these, of course, were initially characterized by Ron Mackay and Paul Teaser, as well as Ludovic Valier. And so the important thing to note here is that this prime stage is really the stage where the cells are ready to differentiate into the different germ layer progenitors. Whereas in the naive stage, if you want these cells to actually form neurectoderm, for example, they first have to transition through what's called the formative stage to the prime stage, which takes about 24 to 48 hours. And that conversion process is quite difficult to control. The cells proliferate very rapidly. They're prone to apoptosis. And so if you have that as the first step of your in vitro directed differentiation protocol, it actually makes it quite difficult to control what's going on. So we thought that working with mouse epiblast stem cells would be a good way to improve the reproducibility of organoid differentiation from mouse pluripotent stem cells. So we implemented and kind of tweaked a bit the EpiSC culture conditions that were out there. We sort of landed on this final setup where we culture the cells on feeders, which unfortunately is still necessary, I think, for the mouse cells. They're an N2B27 with FGF2 and activin A. And the key thing is that we add a Wnt inhibitor, in this case, a tankerase inhibitor, to prevent Wnt signaling in the culture. And this was shown by several groups about 5 to 7 years after the original description of epiblast stem cells to really help prevent spontaneous differentiation, which seems to be more of a problem in mouse cultures compared to human. And so we split these as small clumps. They look morphologically similar to human pluripotent stem cells in the prime state, and they grow quite robustly in these conditions. And just as a quick side note, when we grow EpiSCs this way, they can really be a superior starting point for in vitro directed differentiation studies. And we've shown in the work I'll tell you about that they can make very efficiently cerebral cortical organoids. But in other studies, which are on bioRxiv or have been published recently, We've also shown that you can take epiblast stem cells and very efficiently differentiate them into tissue-resident macrophages and microglia, as well as definitive endoderm and progenitors of the embryonic stomach. So if you want to do in vitro differentiation with mouse, if you start from epiblast stem cells, it is very much easier to generate a pure population of desired lineage of interest than it would be if you started from the naive state. So modeling what has been, or based using as a starting point what has been done for human pluripotent stem cell organoids to generate cerebral cortical fates, we tweaked the timing and concentration of various inhibitors to come up with a protocol to specify consistently cerebral cortical identity in developing mouse epi-SC organoids. So we seed about 1,000 epi-SCs in an agar-roll plate. We add dual SMAD inhibition, Wnt inhibition for the first 48 hours to promote telencephalic identity, and we block sonic hedgehog throughout the first 4 days to maintain dorsal identity. And we found that it can be helpful, though it's not essential, to add a pulse of Wnt activation on between the 3rd and 4th day to help further promote the dorsal identity. So all of the patterning of these organoids happens in the first 4 days. They're embedded in Matrigel from day 1 to day 4, and then they go into an orbital shaker after that in just N2B27 media.
I ask you a question about that Wnt activation step?
Yep.
Just very, very briefly, just a technical question.
Mm-hmm.
So is that, when you say to promote a dorsal fade, is it a dorsal neural telencephalic fade that it's specifically helping? Or is it for other fates as well that Wnt activation step is useful?
So it's a little tricky, and I don't know that we know exactly why that helps. I mean, obviously, Wnt promotes like cortical hem fate as well. If you add too much or if you add it too early, and we had definitely seen that in our previous iteration of the organoids we were making, that we would sometimes get hem like fates. But I think it just somehow antagonizes the sonic hedgehog activity and promotes dorsal, like telencephalic cortical identities. And I don't think we know exactly why that is. But people have seen similar things in human as well.
Could it be dose dependent?
I think it probably is, and we haven't done a significant, a careful examination of that. And so I think, in previous iterations, we had been adding less sonic hedgehog inhibition, and we were getting more ham-like fates. But when we tweak the protocol this way, then that first 48 hours of Wnt inhibition, I think, seems to bias away from generating ham-like identities. But yeah, I think that there's a lot of developmental biology happening there, and in the mouse, it happens very quickly. And I don't think we've parsed that out completely here. So here's what the organoids look like. At day 1, they form these nice EBs, and then you start to see lumens, and they grow over the first week or so. And so very rapidly, we get nice formation of neuroepithelial tissue in these organoids. So this is a cortical organoid at day 4, where you see this nice Nestin-positive radioglial cells that are surrounding an apical membrane in the interior of the organoid marked by ZO1. And you can see that the mitotic, as expected, the cells are undergoing mitosis at the apical membrane. So you can really get quite nice organization of these organoids just in a couple days. If you compare the organoids here on the right to the embryonic cortex in vivo at E11.5, they look very similar in terms of the thickness of the tissue and the expression of these markers. So they seem to do a nice job of recapitulating these early stages of neuroepithelial formation and radioglial cell development. And so importantly, we were able to, in the conditions that we finally optimized, get consistent specification of cerebral cortical identities. And so you can see here that the telencephalic marker FOXG1 is homogeneously expressed in these cells, in these developing neuroepithelial tissues at day 6. And even by day 6, you're starting to get generation of deep layer neurons here marked by CTIP2. You see nice delineation of distinct progenitor domains within these cortical organoids as well, with PAX6 positive radioglial cells surrounding these apical lumens, intermediate progenitors arising slightly more outside of those PAX6 positive radioglial zone, and then TBR1 positive deep layer neurons again on the outside, so migrating radially away from the apical membrane. And so we do see generation of both deep layer and upper layer neurons in these cortical organoids. So here I'm showing you a day 16 organoid. This is just the outer shell, but you can see you have sort of interspersed CTIP2 positive deep layer neurons, as well as brain1 positive upper layer neurons, and their expression is mutually exclusive. And we also see expression of the upper layer marker CUX1 in these organoids as well by day 16. And so we don't only get the neuronal cells that are derived from radioglia, but we also see oligodendrocyte progenitor cells generated in these organoids, as well as astrocytes. And in this case, these are cortical or dorsal telencephalic oligodendrocyte progenitors. So they're coming from radioglial cells. They're not coming from the ventral telencephalon. And so we see by day 16, we've switched from the neurogenic divisions of the radioglia to these the generation of astrocytes and oligodendrocytes. And so we did a lot of transcriptomic and analysis and epigenomic analysis of cells in these cortical organoids. So broadly speaking, we can sort of validate the cell populations that I mentioned and showed by immunostaining, and we can see this nice recapitulation of the radioglial temporal progression from radioglial cells to intermediate progenitors to cortical neurons captured in these organoids. And so we did a bit of a deeper dive into the cell fate specification within the neuronal subtypes there, and we can see some early bifurcation into the different subtypes, broad subtypes of cortical neurons that we would expect to see. So we have deep layer neurons, subcerebral projection neurons, and layer 2, 3, 4 neurons. And so these are markers from Paolo Arlotta's work characterizing the mouse embryonic cortex in using single cell RNA-seq and ATAC-seq. So we see sort of the same broad early bifurcations of cell fates that you see in immature cortical neurons in vivo. And so if we leave these a bit longer, now looking at day 30, we get very nice mature looking astrocytes. Here they're sort of coating the surface, but they're also interspersed in the neural tissue inside the organoid as well. And then we actually see the generation of myelinating oligodendrocytes as well. Here marked by the myelin basic protein marker. So they do continue to mature, and we get nice mature glial cells in these organoids after just 4 weeks. So one of the great and convenient things about this organoid model compared to human is that the organoids mirror the kinetics of mouse embryonic development. And so here is sort of a side-by-side image of our mouse organoids at day 8 and human cortical organoids from Guo Li Ming's lab. And so you can see basically that they, in about a week, our organoids reach the same stage that takes about 52 days to reach in human. So we really can encapsulate a lot of neural development in a relatively short time compared to the human organoids that are available out there. So that's one of the key advantages of this method. And so just quickly, so this is all published in the manuscript, but I just wanted to briefly mention this from the standpoint of reproducibility of this protocol, which is, I think, a very important issue. So we've used this protocol to generate organoids from 16 different iPS cell lines, which come from 4 different F1 hybrid genetic backgrounds. And we perform single cell RNA-seq on each of those 16 lines. And what you can see is that we basically are able to generate reproducibly the same subsets of neurons and astrocytes, oligodendrocytes from each of these lines, suggesting that the protocol is very robust and able to give us consistent specification of cortical identity across a wide range of genetically and technically diverse cell lines. So just to summarize, we have developed a new protocol for generating mouse cerebral cortical organoids from epiblast stem cells that's now been validated on greater than 20 cell lines. These organoids recapitulate key features of the embryonic cortical development, including the cellular architecture, cell fate specification programs, and developmental timing. And we think this model will be a valuable complement both to mouse in vivo studies and human in vitro organoid studies when you're examining mechanistic aspects of cell fate specification and gene regulation. And we're continuing to try and improve this protocol and work on conditions that enable longer term maturation of these organoids so that we can study hopefully somewhat later stages of development. So that's sort of the end of the first part. And so then the second challenge that we really needed to address was to have access to genetically diverse pluripotent stem cell lines that would allow us to perform systems genetic studies of the developing brain. And so this project was led by Rachel Glenn, a graduate student in the lab who graduated earlier this year, together with Stephanie Doe and Raghavi Vijayakumar, who's a grad student in the lab who's taken over and has done some of the expression QTL mapping studies that I'm going to talk about. And Yas Faruta, who's the head of our mouse genetics core at Sloan Kettering, played a critical role in this as well as Mike Coons, who's a bioinformatician that we worked with in Germany, and Kushal Day and Kartik, who are statistical geneticists, and Richard Koch, who's at the Center for Epigenetic Research at Sloan Kettering. And this is all described, most of this is described in a preprint that's been on bioRxiv for about a year now, as well as in a companion paper in Stem Cell Reports, which I encourage you to check out. So diversity outbred mice are an amazing resource for systems genetics that's been developed over the last 20 years through the work of the Collaborative Cross Consortium, which encompasses investigators from Jackson Lab, UNC, and several other universities. And so the point of this population was to generate a genetically and phenotypically diverse stock of mice that could be used for genetic mapping studies. And in order to do that, basically, they took 5 inbred strains, which are what are sort of referred to as common laboratory strains, which come from Mus musculus domesticus, and then also 3 wild-derived inbred strains, which are much more genetically and phenotypically distinct compared to the standard lab strains, and actually come from different subspecies of mice, which have diverged from standard lab mice by 500,000 to 1 million years. And so they did extensive intercrosses between these different inbred strains, and they've now been doing that for over 60 generations to generate this highly recombinant population of outbred mice, which can be ordered from Jackson Lab. And so they each individual is genetically distinct, but they contain alleles that all come from these 8 distinct inbred progenitor strains. So this outbred stock captures about 90% of the total genetic variation that's present in available inbred strains of mice. There's approximately 45 million segregating genetic polymorphisms And the genetic architecture of this stock enables high resolution QTL mapping at relatively reasonable sample sizes compared to what would be required for a similar study in humans. And they've been widely used for mapping quantitative trait loci, influencing a variety of behavioral, anatomical, and physiological traits, mostly focusing on adult animals. But they, have become a go-to model in toxicology and some other phenotypic studies because they exhibit such substantial phenotypic variability, and they nicely model sort of the variability in responses to, for example, a drug that you would see in the human population. So a key feature of these diversity outbred stock is that compared to humans, the average allele frequency within this population is quite high. And this is a key parameter for genetic mapping because if the allele frequency is too low, if you have 1,000 individuals, you might only have a few individuals out of that 1,000 that even carry an allele of interest. And so, for example, if you compare the humans in the Human Genome Project, the vast majority of SNPs that have been identified in those 1,000 people are present in less than 5% of the total people in the population. Whereas in the diversity outbred mice, nearly all the SNPs are present at a frequency above 5% in the total population. So it's much easier to, basically most of the SNPs are common SNPs, and that allows for that's very convenient for the sake of genetic mapping. So diversity outbred mice exhibit, sorry, can you hear me? Oh, sorry,
Yes.
can you hear?
Yep.
Okay,
Yes.
sorry, yeah, I touched my headphone. I apologize that it muted me. So the diversity outbred mice exhibit extensive phenotypic variation. And so this is a study from across the street at Rockefeller University from Priya Rajasethupathy's lab. So they just were interested in looking at behavioral variation in diversity outbred mice. And so they have a short-term working memory task that they use to study short-term memory. And what they are looking at here is if you look at C57 Black 6J mice and you put them through this behavior, you get this is sort of the variation in behavioral output that you get. Whereas if you then do the same behavior, but you incorporate a diversity outbred mice, you can see that there's a broad range of behavioral variation that's not observed in the standard population. And they were able to use this to map some quantitative trait loci that can potentially contribute to these differences in working memory. So a nice thing about the diversity outbred stock as well is that we also have access to the original inbred strains that were used to generate the stock. And so then what you can do as a starting point is assess variability between those 8 inbred strains as kind of a quick survey to see how much variability you're going to get when you have a larger panel of cell lines. And so with assistance from Laura Reinhold at Jackson Lab, she gave us 2 embryonic stem cell lines from each of the 8 founder strains that were used to generate the diversity outbred stock. And then we converted those into epiblast stem cell lines and performed RNA-seq and attack-seq on those. And we also differentiated them into definitive endoderm and performed similar experiments. And one thing I should note here is that in general, we've had a great success converting embryonic stem cell lines into epiblast stem cell lines in vitro. It's just a few day process. There was one strain here called AJ, which we were not able to stably culture as epiblast stem cells. And I'll get back to that in a bit as to why we think that might be. So if we look at gene expression for pluripotency markers broadly across these different inbred strains, we can see that in these culture conditions, we can generate prime pluripotent stem cells that express the core features of the prime pluripotency gene regulatory network. They have low expression of naive pluripotent stem cell markers, and we see always a bit of spontaneous differentiation in some of these lines. And so we do start to see a little bit of primitive streak associated genes, but generally speaking, there's a strong core prime pluripotent population in each of these from each of these strains. I'm not going to get into this too extensively, but basically when looking at the transcriptome and the epigenome, these cells cluster by their strain of origin predominantly. And so we have a very extensive characterization of transcriptomic and epigenomic features of these cell lines, from each of these inbred strains across multiple clones of cells as well. So one sort of interesting and obvious thing to look at was to ask, well, we know that they have differences in gene expression. Does that contribute to functional differences in their ability to differentiate into specific lineages? And so we had developed a very efficient protocol to differentiate epiblast stem cells into definitive endoderm. It's a two-step protocol that takes about 40 hours. And so we applied this protocol to each of those 16 or so epiblast stem cell lines from each of the inbred strains to ask how the efficiency of differentiation varies between different inbred strains. And so I really like this data because I think it's really informative. And so we had optimized the protocol on this inbred strain called DBA. And so you can see we get very high efficiency definitive endoderm from across multiple cell lines and multiple technical replicates from this line. And when we look at several of the other lines which are closely related to DBA, we can see similar behavior in this differentiation assay. But if you look at the outbred or the wild-derived strains that are part of the DO, such as Castaneous and PWK, the efficiency of definitive endoderm differentiation is definitely reduced, and it's relatively consistent phenotype across different lines and clones. So we think that this, standing genetic variation between these different inbred strains influences not only their gene expression and epigenomic features, but also their functional capacity to differentiate into different cell types. So given that we had observed these interesting phenotypic differences, then the next step would be to try and actually map quantitative trait loci that might be contributing to those differences. But as I mentioned briefly before, this quantitative trait locus mapping requires relatively large sample sizes. And so here is actually a power calculation done for looking at QTL mapping in the DO stock. And so you can see that when you have 200 cell lines, you're able to relatively confidently identify variants that contribute to at least 20% of the total variation within the population. Which is a good amount of power for an eQTL study, for example. But if you go up to 400 cell lines, you can really start to map variants that explain even as little as 10% of the total variation. And so this compares favorably to the sample sizes required for human quantitative trait locus mapping, but it still presents quite a challenge because most of the experiments that are done in stem cell biology are from a relatively small number of cell lines, like 2 or 3. And so we needed a way to generate a panel of cell lines that could be used for these large scale studies. And so in order to do that, we, working with Yas at the Mouse Genetics Core, we came up with a strategy that we call pooled in vitro fertilization. And so effectively, it's the same as normal in vitro fertilization, but instead of having one male donor and one female donor, we have between 10 and 15 males and 10 and 15 females. And then we perform these pooled IVF drop reactions and then directly isolate blastocysts from the fertilized embryos. And from those blastocysts, we can directly isolate ES cells. And so we've done 4 rounds of this, each round with 10 to 15 different males and females. We bank the ES cells at a very early passage, so between passage 2 and passage 3. We then expand them one more passage to have a larger working stock. They're genotyped using a genotyping array that's available in the field and was designed for this mouse stock. And then we convert them to epiblast stem cells, which then by the time they reach the epiblast stem cell frozen stock, they're about passage 6 or 7 total. So, these are very low passage pluripotent stem cell lines compared to what most people in both the mouse and human stem cell fields work with. So we think this is a nice advantage of this mouse model in general, is that we have ready access to these kind of very fresh and new stem cell lines. So in total, we've done this 4 times and derived about 250 total ES cell lines. After performing QC assays on them, we had about 230 that passed QC, meaning that they didn't have an obvious chromosomal abnormality based on pseudokaryotyping from the genotyping array. And then we were able to convert, of those 230 ES cell lines, we were able to convert 229 of them to epi-SCs. And one thing I should note is that historically in the mouse ESL field, most studies are performed with male cell lines, because female cell lines have some epigenetic instability in the naive state. But when you culture cells in the prime state, we're able to stably culture both male and female cell lines. So we have 130 males and 95 female cell lines in this panel. So from the genotyping data, we can impute genotypes for each of the cell lines. This is an example of one of the cell lines, what they look like. You can see they are highly recombinant and have alleles from each of the 8 founders. We can map these genotypes in PCA space, and we can see that from each batch of IVF that we generate genetically diverse lines that are sort of mostly non-overlapping. And importantly, for 8 of the cell lines, it's not feasible to differentiate all of the cell lines into each of these lineages, but for 8 cell lines, we tested their ability to form the 3 germ layers, and each of the 8 lines can robustly be differentiated into definitive endoderm, mesoderm, and neuroectoderm in vitro, suggesting that they're functionally pluripotent. And one thing to note here, which is an interesting comparison to what I showed previously with the inbred strains, is that generally speaking, these outbred cell lines have performed better in this directed differentiation assay than the inbred strains did. So we get more consistent results. So here, this is 8 different cell lines, 4 independent experiments of differentiation. You can see that there's a relatively consistent percentage of definitive endoderm generated from each line. And broadly speaking, it's relatively high compared to the inbred strain results that we got previously. So, based on work from Michael Wells, Steve McCarroll, Kevin Egan, and others, we thought that in order to really be able to do anything with this large cell line panel, we would need to use multiplex cell culture, or so-called cell villages, because it's difficult even to perform basic QC across 200 cell lines to be able to do anything. And so as a pilot experiment, what we did was we generated pools of 6 lines. I guess Mike can weigh in whether that's a village or not. Maybe it's a town, or it's not big enough to be a village yet, but so we made from a total of 150 unique DO cell lines, we made a panel of about 60 pools of 6 lines per pool. Then those are banked and frozen down, and we thaw those and then combine 5 pools into 1 lane for 10x sequencing. So only 6 cells are co-cultured together, but then groups of 30 cells are processed for single cell together and run in the same 10x lane. Then using the dropulations pipeline from Steve McCarroll's lab, we can genetically demultiplex those pools, so we can identify which individual each cell line came from. And then we can use that data to perform single cell expression QTL mapping, and also in theory map cellular trait QTLs that we from phenotypes that we can infer from the single cell data.
Tommy, is this all in the epiblast stem cell state?
Yes,
In this
right now,
stage?
yeah. Well,
Okay,
yeah,
great.
I'll show you a little bit. So there's a little bit of spontaneous differentiation too. So we capture a little bit of primitive streak and definitive endoderm as well. But the goal here was to have, to focus on the epiblast stem cell state.
Wonderful. Thanks.
So an important technical note here, and so something that's obviously been an issue in the human cell villages field as well, is that we do see after the initial pooling, freezing and thawing, there is some differences in representation of the different cell lines. So these are ranked in order of the most abundant cell line to the least abundant cell line. So generally speaking, we can recover 5 or 6 of the cell lines from each of the pools consistently, but there are definitely quantitative changes in the number of cells in each pool. And so for things where you're not using single cell RNA-seq as phenotyping, that could potentially be problematic. But generally speaking, we can recover enough cells from each experiment, from each cell line, that it's not too much of a problem. And we've done no optimization to try and tweak parameters to be able to make that better. So I think there's a lot of room for improvement still. So we have in total from those experiments, we have cells from 139 donors and about close to 160,000 cells that passed QC and were demultiplexed successfully. So you can see, broadly speaking, that there's the main clusters of cells express markers of prime pluripotency like OCT4 and OTX2. And then we also have these sort of slightly primed cells which start to express FOXA2, and then more primitive streak-like cells that express brachyury. And then at the bottom, there's a small subset of cells which are definitive endoderm. And so we do capture a little bit of that primitive streak to definitive endoderm differentiation trajectory in these experiments. So if we look at, we assign basically each of the clusters to an identity. And so here, there's a variety of clusters which represent epiblast stem cells. And to some extent, things like cell cycle are distinguishing these clusters, but broadly they express markers of, I'm trying to move that out of the way. Broadly, they express markers of prime pluripotency, limited expression of brachyury and other markers of differentiation. And so we were able to pull out clusters that we think represent epiblast stem cells. And we can see essentially those other clusters that represent some of these spontaneous differentiation fates. So one thing we were interested in, because there actually is not a lot of single cell RNA-seq data from mouse epiblast stem cells, was to look at molecular variation and whether we see differences in primed in pluripotent states, like for example, formative versus primed, or kind of an anterior epiblast-like or posterior epiblast-like fate. And so there was a recent paper from Elisabetta Ferretti's lab looking at mouse epiblast in the anterior and anterior posterior genes that define anterior posterior axis in mouse epiblast. And so if we look at some of the markers that she described as being biased towards the anterior epiblast, we do see that there's clusters that seem to have higher expression of those markers. And similarly, they express higher levels of OCT6, which is a prime pluripotency marker. And then there's clusters that have higher expression of markers that are more indicative of more posterior epiblast-like fate, such as FOXA2 and TRH. So we do think that there's some pluripotency differences or differences in state that are present in the cultures, and that's something we're continuing to try and define more. It's not completely trivial, I will say. Okay, so then we wanted to look at not only So one thing we noticed immediately from looking at these different cell fates was that there seemed to be differences in the rate at which different donors, cell lines from different donors spontaneously differentiated. So here's the fraction of cells in blue that are we defined as epiblast stem cells from each individual. And you can see that for some individuals, almost all the cells are undifferentiated, whereas for other individuals, they have a more significant population of primitive streak-like or definitive endodermal cells within the culture, suggesting that their behavior in kind of the same culture conditions might be different. And so we pseudo-bulked the EPSC clusters together for the purpose of EQTL mapping. There are fancier ways to do EQTL mapping at the single cell level than that, but initially we thought it would be better to focus on the pseudobulk transcriptomes. So we have pseudobulk transcriptomes from 130 individuals. You can see, broadly speaking, they express high levels of prime pluripotency markers, although there's definitely some variability in the levels of those markers between cell lines. So then we wanted to map expression QTLs from this panel of genetically diverse EPSCs. And so using RQTL2, we were able to map approximately 4,000 E genes that are modulated by genetic variation. And these, included genes from a wide range of signaling pathways that are involved in canonical pluripotency, as well as genes linked to cell attachment, apoptosis, and other things that might be of interest for the behavior of these pluripotent stem cell lines. And so, for example, over 60% of genes in the FGF signaling cascade have significant EQTL hits. So the genes in green here have greater than one significant EQTL associated with them. So we see throughout these signaling pathways at all levels, from the extracellular receptors to the nuclear transducers of these pathways, we see variation in their expression levels across these different cell lines that are modulated by genetic variation. So here's an example of that. KRAS, important intermediate in the RAS-ERK pathway, seems to have a strong expression QTL associated with it in cis. And so you can see these three strains, having this genotype at the KRAS allele leads to much lower expression of KRAS. And we could validate that actually in the parental gene expression from the parental cell lines, where we see consistent changes in RAS expression in those same parental cell lines. And here's an example of what this data looks like. So we have lines that are homozygous for the variant allele versus lines that are homozygous for the common allele. You can see there's a very significant change in their expression of RAS. Sorry. Then we also wanted to look at not just molecular QTLs, but also functional QTLs. So we wanted to see if we could map quantitative trait loci that impact the frequency of spontaneous differentiation that we observed between the lines. And so using as input the data from here. So this is the input for this QTL mapping. Okay. We were able to map 3 suggestive QTLs. And this one on chromosome 3 was particularly interesting and was really close to approaching the level of significance at this sample size, we were happy because it's quite difficult to map these transacting QTLs in pluripotent stem cell populations. So this was actually exciting because when we looked at where this is in the genome, it's actually the center of that window is SOX2. So a core regulator of pluripotency, suggesting that there's differences potentially in SOX2 function or regulation that might be modulating the frequency at which cells differentiate. And so if we look at the allele effect plots, so this tells us which allele is associated with the phenotype. Interestingly, AJ and WSB strains seem to have a higher, their allele is associated with a higher frequency of spontaneous differentiation compared to the other inbred strains. And so then when we looked at the expression QTL data, we could see actually that there was this very nice and consistent change in expression of SOX2. So those same strains that have a higher propensity for differentiation have lower expression of SOX2 in the pluripotent state. And this was actually similarly, we could see the same trend in the parental inbred strains. And AJ is not here because, of course, we couldn't culture those lines. So
can I ask you a question about this? So when you say differentiation here, you mean mesodermal or endodermal differentiation, do you?
So we took basically the fraction of cells that are undifferentiated over the fraction of over the differentiated cells. So like it's basically the differentiated cells pretty much exclusively consist of primitive streak-like cells and definitive endoderm.
Right. Perfect.
So we're not distinguishing within that differentiated subset. We're not mapping the frequency of primitive streak versus definitive endoderm. It's just total undifferentiated versus total differentiated.
Yeah.
Yep. So this was interesting, and as I mentioned, it's exciting because it links back to one of our initial observations, which is that the AJ inbred strain, the epiblast stem cells from that strain really do not grow well in the culture conditions that we use, which have worked for 245 out of 246 other strains. And so we think this SOX2 change in SOX2 regulation might be part of that. And we're trying to understand more about the molecular mechanisms at the SOX2 locus that might explain that. So looking at things like structural variants and SNPs that are present on those haplotypes to see how they might modulate SOX2 regulation. And then I think very quickly go through the last part. So we're probably want to save some time for a discussion. So of course, we don't want to just look at pluripotent stem cells. Ultimately, we want to be able to differentiate those cells, ideally in the context of villages, to be able to map molecular and cellular QTLs in neural tissues. And so Raghavi has been working on developing a scalable way of differentiating epiSCs into telencephalic neural progenitors. Ultimately, we want to do organoids in the intervening period. Having a neural progenitor platform is sort of a nice mix of getting some neural identities, but also not having it be as complex as an organoid. And so what she did was basically took the protocol we use for organoids for cortical organoids and basically stopped it after 3 days and split the cells, dissociated the cells, and grew them in 2D in EGF and FGF2 for expansion. So they're patterned to a telencephalic fate in 3D, which we think is very helpful because it helps control the density of the cells while they're being patterned. And then they're dissociated and cultured in EGF and FGF. And she's generated bulk transcriptome data from 70. She's converted 70 DO cell lines using this protocol and generated bulk transcriptome data to try and understand how variable they are. And so what you can see here is that there's a nice conversion into SOX1 positive, FOXG1 positive neural progenitor cells, and the percentage of cells that are positive for those markers within these cultures is relatively high. They can differentiate robustly into neurons, so deep layer projection neurons that are TBR1 positive, astrocytes and oligodendrocytes, suggesting that there are multipotent neural progenitor cells. And then if we look at markers of using the transcriptomic data, they express consistently anterior forebrain markers such as OTX2 and FOXG1. And broadly speaking, they seem to be dorsal. It's that they have a much higher expression of PAX6 than, for example, NKX21. This is true across most of the 70 cell lines. So FOXG1 is in the middle here. So most of the cell lines have robust expression of FOXG1, suggesting that, broadly speaking, this protocol can successfully differentiate the vast majority of these cell lines into a dorsal or a telencephalic identity with some variation in the dorsal ventral axis, which we're still trying to parse out a bit. Raghavi's also used this to map EQTLs in those NPCs. We're still sorting through that data as well, so I won't tell you about that. And just very briefly, can summarize some of the things that we're hoping to be able to do with this platform in the coming years. So we now have 250 cell lines from the DO mice that we can work with and potentially pool together. We want to be able to differentiate them ultimately into organoids in either a pooled or arrayed format, which will allow us to look at a developmental time course of cortical development in vitro. And then we'd like to be able, the same way we mapped expression QTLs with single cell RNA-seq data in pluripotent stem cells, we'd like to do that in the organoids and look at things like cell abundance QTLs, as well as pathway or program level QTLs in the gene expression data. And, of course, an interesting kind of next step beyond that is to look at how these different genetic backgrounds modulate the phenotype of neurodevelopmental disease genes, because there's excellent studies looking at how the phenotypic penetrance of neurodevelopmental disease genes is modulated by genetic background in mice, in actual live mice. And so there's a lot of reason to think that there's a lot of molecular modifiers of disease genes that are differentially expressed across these different strains that we can map using QTL approach. And so we'd like to combine CRISPR-I, be able to perform CRISPR-I in these village populations to look at how genetic background is influencing disease gene presentation in these organoids, and ultimately to hopefully be able to map as a way of scalably mapping molecular modifier alleles for these disease genes, which could be used to suggest therapeutic approaches. And this is work that we've been starting to pilot with Tom Norman's lab at MSK. So I acknowledge people doing the work along the way, but these are all collaborative projects that everyone in my lab was involved with. So Danny was sort of leading the organoid work, and then Rachel, a former grad student, led the diversity outbred studies, and Raghavi, a newer student, has been working and doing the QTL mapping studies in neural progenitor cell work. And we've worked with a lot of collaborators, especially Laura Reinhold, Chris Baker, Steve Munger, and others at Jackson Lab, who've been very generous with their expertise working with DO mice and some of the issues associated with that. So thanks for your attention, and I'm happy to have some discussion about our work.
Thanks so much, Tommy. It's very cool to see how much effort and patience you put into generating the protocols, the lines, and then the cell stocks to be able to ask so many questions going forward. So I'm very happy to open up to the questions from anyone. Jump on in.
I have a question.
Hey,
great, Flora.
Hi. Since you've done such a nice characterization of the stages of differentiation, I wonder whether you have done any comparison between mouse and human in terms of gene expression, but also proportion of different cell types?
In the pluripotent stem cell cultures or in the organoids?
Both, I guess.
Yeah. Yeah, so we've been...
It's not an easy experiment, so maybe you can't do it in both, but to the extent that you've done that at any stage would be interesting.
Yeah, we've been really, so we talked about this a little bit before the meeting started. Martin Pera and others have characterized sort of subsets of substates within standard pluripotent stem cell culture conditions that have sort of more formative or primed characteristics. And that sort of broadly corresponds to either early and late mouse epiblast or anterior and posterior mouse epiblast populations. I think it's not so easy to take the in vivo epiblast data and put it into in vitro 1-to-1, because I think there are broad differences between in vitro and in vivo. So we've been trying to merge together what's known about substates in human cultures with what's been characterized in mouse epiblast at different stages and in different regions of the epiblast, and trying to see how that varies in our cultures, and then compare also the mouse versus human. There's sort of a broad idea that human pluripotent stem cells are more formative like than mouse. I think that's maybe a little bit of a dated perspective, because I think historically, if you culture mouse epiblast stem cells without Wnt inhibitors, they tend to be more differentiated and then thus look more primed than kind of the human cells. But I think it's a very interesting to look at these cross species differences. And I think that's something we really want to dig into more. And we're sort of trying to figure out the best way to do that. Because at the single cell level, it's actually not trivial to parse some of those like formative primed continuum questions. And historically, it's been done more at the bulk level or with sorting for specific markers. So we're trying to kind of translate that into the single cell data. It's not completely trivial so far.
Thank you.
Wow.
I can jump in. I'm very interested. You showed that there is a different proportion of donors when you pull the lines. If this does correlate actually with the differentiated or undifferentiated states of lines, because we have tried to find correlations to design pools that better work, and this it's not showing in our data, but I was wondering in your case if that's the case.
Yeah, so we need to go back and look at the dropout rates and see if those lines have alleles that suggest that they would be more primed for differentiation. We didn't do that in the initial differentiation mapping, because yeah, you're right. Like if the line is differentiating too much, it's not going to be maintained, and it's going to be outcompeted by the epi-SCs that are pluripotent over time. So I think that's one of several possibilities as to why certain lines drop out and others don't. I think things like cell adhesion, survival when they're dissociated, survival when they're frozen, survival when they're rethawed. We try and minimally manipulate them and grow them together as little as possible to minimize that. But I think there's probably additional things that might be necessary. Like we don't use ROCK inhibitor and things like that for standard culture, and I think things like that might help. But yeah, we should definitely look at whether the lines that drop out are, they have alleles that might be similar to the type of alleles that we find in the spontaneous differentiation QTL mapping that we did.
Jump on in, Michael.
Yeah, I just want to follow up on something. First of all, this is a great talk. I really enjoyed seeing all this in one place. Because I've been following all the preprints.
Oh, thanks.
So it's actually about the same thing. This is more of, I can tell you what we're doing over here. We're not differentiating as villages. So that's a very important caveat to what I'm about to say. We have noticed over and over again that the arrayed growth rates, so the growth rates of these lines when they're cultured separately, correlates very well with their growth rates once they're a village. And so we're actually working on a model that we're calling optimized seeding, where we will plate each donor at a different seeding density, different number of cells based on their arrayed growth rates that we're actually calculating between thawing them, expanding them, and pooling them. So we actually, over those 5 or 6 days, we know their real-time growth rates by taking pictures every day on a bright field and running it through CellPose. And then what we can do is say, okay, this donor is growing slower. We need to add, instead of it being 1% of the village at day 1, it's going to have to be 2%. And this one's growing super fast, so we're going to lower it down to 0.5%. So we're going to, I was just discussing this experiments this morning with Harold Pimentel, who's the one who's designing these bioinformatic tools. And so we'll probably have this thing wrapped up by March or April, and we're happy to share what we found with everybody, because this is the most consistent problem people have, is how do you keep them balanced? And the idea would be, hey, I want to harvest on day 7 after pooling. What should be my seeding density for each donor if day 7 is my goal for when I'm harvesting for these experiments?
No, that's really interesting. Yeah. No, thanks for sharing.
Another question I had is like, these growth rates is referring to the differentiation themselves or just maintaining them at ideal stage? Because I think that depending on the fate, this might change. So it might not be the same for the endoderm or the cortical one.
Yeah, so that's what I want to make sure it's very clear. We're not different. So we're all working with NPCs in my lab. And so it's a in the array they're NPCs, in the village they're NPCs. We're not differentiating them. So that's the big caveat to this as a potential solution is it's really hard to predict how they're going to differentiate and how quickly and all those things that Tommy was talking about earlier. So this won't solve that problem yet, but we're hoping it's a good first step for those who are trying to at least get their stem cells or their NPCs to play nicely with each other prior to any sort of differentiations.
Well, you also have to consider that you're forcing the cell in a very unphysiological state. Cells are foreign to each other. They don't have the same surface molecule, cell adhesion, immunological characteristics. So this is very difficult, I'm sure. And I wonder whether competition could be much less if they're not exposed to their own sort of so intimately exposed to each other that way, It's very difficult.
Following on this, there is a paper I've read recently which says that actually Wnt activations facilitate that there is less cell competition, but we need Wnt inhibition if we weren't forebrain neurons, right? So there is a balance between what you can do and what you should do.
Yeah, we were very interested to see if there would be cell competition phenotypes in our cultures, because it would be actually very interesting to map alleles that might modulate cell competition, because it's kind of this confusing, multifaceted process where the exact molecular players are not completely clear. We haven't been able to definitively find that, and that sort of gets at Michael's point about the growth rate. If the independent growth rate is a predictor of the growth rate in a pool, that would suggest there's not very much competition happening. But when those numbers differ, it'll be very interesting to see what you guys see, once you're able to modulate that more carefully, whether there are kind of lines that behave in a way that is not expected, I guess, from the individual growth rates. Because that I think might point to some kind of interesting biology happening in terms of their epithelial biology or the way that they're interacting with one another.
Yeah, definitely. When we culture, we actually try to minimize the physical contact by culturing at much lower densities, but that only works until a certain point, right? These cells are growing, they're proliferating. And so at some point, if you want to go a prolonged period of time without a passage step in between, we're going to have to figure out how long we can go before cell competition does or does not kick in and start really messing with things.
Okay, did you have something to jump in
Yes,
with?
can I jump in? Hello, Thomas. This is great work. Thanks for all your effort. I think it has a great potential to applicable for many approaches. So, my
thank you.
question is, I think no, you already have some plan, but I think to understand the genetic background, QTL analysis to certain phenotype, it would be nice to have a specific condition, the known phenotype that you create, and then correlate with the genetic background with that. So what I mean is, if the question is about the neural, lineage specification for the definitive endoderm, you can ask for more specifically for the activin signaling, right?
Mhm.
What I'm saying is we might need to have a activin treatment condition and also activin inhibition condition, and then cell will behave differently. It will show the phenotype difference much more clearly, and then we can easily link with the genetic difference, right?
Yeah.
So I think right now I think you did just undifferentiation gene profile, right? And then neural stem cell condition that you have the data set. And I think that's the system, and I think maybe you are thinking about specific treatment or condition that that you're gonna use with this stem. So I want to hear what is your plan, or do you agree with this idea?
Yeah, no, I think you bring up a really important point, which is that, I think your ability to find phenotypes in these models is very dependent on exactly the conditions that you are looking at and how you design the experiment. And we've been sort of still at the kind of proof of concept setup validation stage, as I think many people are, because it's sort of a big undertaking to get things going. But yeah, I think the hope is that if we have specific questions, that things like response QTL mapping, which I know Michael's lab has done, and I think he just dropped off the call, but
Yeah, there you go.
yeah, but so then the thought is that those kind of like response phenotypes or like acute signaling response phenotypes might have a simpler genetic architecture too than some of these like more ambiguous differentiation phenotypes. I will say though that another thing to think about though is that a lot of the ways that we do directed differentiation, we're providing a very strong differentiation signal to the cells, and the goal is to actually have them be as homogeneous as possible. It is possible that under those conditions that you're washing out the signal, the subtle differences in these
Different,
lines.
right?
Yeah.
No doubt.
And so then you get in this situation where it's like, well, okay, should I use a slightly suboptimal differentiation protocol to and enable cells that are maybe responding differently to
go a different direction, right?
manifest that difference to manifest a bit more. And that's actually, I think, one of the biggest challenges in this field. But there's a bright side to that, which is that if you want to look at late stage cortical neurogenesis across different genetically diverse lines, you need to get them all to that point. Right. So if you can come up with differentiation protocols that are give very homogeneous results, even from genetically diverse donor cell lines, then that opens up the possibility of looking at the influence of genetic variation on cellular phenotypes at later and later stages of development. So there's like, I guess, like a glass half full, glass half empty way of looking at that problem. Is that you need to control the impact of diversity on differentiation sometimes, and for a lot of like cell replacement therapies, clinical applications, that's really important. But then other times, if you want to actually study the diversity and use it as a perturbation to a biological system, you need to have a system where you can allow that those more subtle phenotypes to manifest.
Absolutely.
Yeah.
Right.
Absolutely. If you guys
Can I
have
pursue that? Go for it, Carl.
jump in, Ron.
Well, I was just going to pursue that question with respect to AJ.
Mhm.
So the AJ line, and that SOX2
the regulatory element. Yeah.
Yeah.
Mhm.
Has anybody gone and looked at AJ embryos to see whether that differentiation difference that you define in vitro actually occurs in the animal itself? That it's an adaptation to some feature of AJ biology in vivo. Is that a sensible question?
Yeah, definitely. Yeah. No, I mean, I think, well, obviously those mice, are mice, and they're not, embryos that resorbed. So they are able to develop normally. I think, so I talked with Kat Agisnakis, my colleague, who's sort of an expert on early embryonic development, about this. I think one tricky thing is that development is so dynamic at this like epiblast stage, right? That
Right.
it is quite difficult to really reliably define like a differentiation phenotype that's somewhat subtle. And because there's a lot of regulative features of development at that stage as well. So that we should really look. We have another. I didn't discuss it today, but from looking at F1 hybrid cell lines, we've also found a strain where there's a very divergent regulation of NANOG in the primed state. And so we've been trying to think about ways to assess that in vivo in embryos as well. And so, yeah, I think we should definitely, it's a lot easier to do it in the AJ strain because it's a common strain that's been used for a long time. So it's a, robust inbred strain that we could get embryos from. But yeah, we should check and see if there is a difference in vivo, because if there isn't, it would suggest that there's compensatory mechanisms,
canalization
maybe.
effects. Yeah.
Yeah.
I'll ask another question about this with this image up. So these wild-derived mouse strains, you said in the talk that they're a million years old, which I find very interesting. So would it be possible to imagine, I mean, without wanting funds or anything, that you could extend this to other mouse species and maybe even to rats? So as I understand it, mice and rats diverged 20 million years ago. So you could really cast a very specific net across species evolution this way. Is that
that's a great
something
question.
you've thought about? Yeah.
Right. No, we have a lot actually, because the one thing that's very nice about this primed pluripotency conditions with Wnt inhibition, and this is something Jun Wu's lab has done a lot with, is that it works very robustly across a lot of different species. So Jun Wu's lab has done horse, cow. Austin Smith has shown that cow also works, like independently shown that cow epiblast stem cells grow well in these conditions. We've made rat epiSCs that work very nicely in these conditions as well. And we've tried to push so these are two inbred strains that are from a different subspecies of mice. So like Mus musculus and Mus castaneus are subspecies, whereas like differences, there are inbred strains that actually are different species of mice that can still, under artificial conditions, interbreed with standard lab strains, such as like Mus spretus is the one that has been used historically for genetic mapping studies. And so we have done F1 hybrid. We made organoids from those, like F1 hybrids of spretus and black 6, and we're doing now more detailed characterization of the epiblast stem cells and the differences in cis regulation that are present. The biggest barrier to that, honestly, is the genomes for all these wild-derived strains have only recently been like high-quality genome builds have been available for the last 2 years or so. And so now you can actually look at structural variants and a much more higher quality genomic data from those wild strains too. So I think we're thinking a lot about that in the evolutionary context as well, because this prime state does allow, like naive state is a kind of divergent between rodents and other species in some ways, but the prime state, you can, I think, start to think about like these large scale evolutionary comparisons and also things like making organoids from those prime cells across different species. I think it makes it more accessible as well.
That's very cool.
Do you know, can I ask another question? Do you know that these phenomena are cell intrinsic? Because if they're not, and especially when you're talking about, factors like Wnt or FGF and things like that, they're likely floating around on the cell surface, right? And we all know these molecules are very much tightly bound to the membranes. So wouldn't that interfere with the mosaic approach? Because all of a sudden you're exposing two membranes very different to each other, putting them in contact, right? Wouldn't that confuse the cells a little bit? Or I'm not sure I can formulate the question, but basically, when you make these hybrid organoids, you're putting in contact two cell surfaces that are not the same, right? And they are supposed to be where they harbor the signaling that regulates a lot of these phase transitions.
Yeah, no, 100%. So that's a very good point, and I think an important thing that needs to be considered in the interpretation of every experiment that's done in these pooled cultures. At least in the in the prime pluripotent state, like Joseph Powell's lab has looked at this more carefully, but and we've looked at it a little bit, but not as extensively as they have. But they basically showed that, the behavior of an individual stem cell line within a pool is largely determined cell intrinsically. So they did that by comparing the same cell line mixed with other, different combinations of cell lines, and they were not able to see, the vast majority of the variation was driven by cell intrinsic differences. I think partly that's because you're averaging across a large number of cells that are, in different, in apposition to a variety of different neighbors. And so there certainly is the possibility that there's non-cell autonomous effects. And I think exactly how big those are and when they manifest is something that's very interesting to try and, think about and figure out the best way to understand that.
So we did an experiment where we tried the usual mosaic approach, as opposed to an embryoid bodies mixing approach, where you keep the embryoid bodies individually formed, but then you mix the embryoid body in the same culture, and the competition was hardly there that we saw when the same lines were mixed up together, in a true mosaic. So I suspect that the cell-to-cell contact is very important in driving this phenomenon where cells outcompete each other. I don't know what's going on. No, I mean, be clear. I'm not sure I understand it, but that's the result.
Two quick notes on that, because we have done the same baseline versus a kind of pool approach. And I think we can be quite sure nowadays, because many labs have found the same, that transcriptionally, when we look at non-cell autonomous effects on the transcriptome, we don't see much differences between the baseline and the same line in a pool. But in other omics layers, I'm not so sure. So I think that's something that we need to explore. And then regarding the non-cell autonomous effects in differentiation, I think that's something we would need to check, because many studies have just focused on the initial stages, and we don't know if the cell-to-cell contact might be important later on. So we don't know. And last one thing, the problem of the imbalance, which could be an artifact of these non-cell autonomous effects. Usually for us, it's very visible just after induction, the 3-4 days after induction. Later on, it's quite stable, probably because cells acquire different identity. But the first 3-4 days is when things can really go wrong for some reason. We don't know why.
Well, isn't the simplest way, or at least for me, the simplest way of thinking about that is that's when the cells are going through these completely non-linear transitions, which we call, amongst other things, organizers. So if you get a kind of a bias in that computation, you could end up in a very different place. I don't know. That's the way I sort of view that early effect. Anyway, it's very cool. This discussion could go on and on. It's wonderful to talk this way. Can I ask
you guys have one more minute? Ron, do you think it'd be interesting to show them what we looked at previously?
Sure. Yeah, sure. Absolutely.
So to give us some insight into these early biases that you've focused us on, and to give us a way of trying to not only think together, but to get data to kind of drive that discussion. Earlier, just before the session, Ron and I were looking at this figure in which you do some differential expression of the wild-derived lines, or all the lines, compared to BLAC6. So there's a packet of genes you show that are higher in the Cas lines, and then, for instance, here, a packet that are higher in the PWK lines, or all three of these. So that would be these two gene lists here. And this is all in epi stem cells. And you did the RNA sequencing. And so here are genes that are consistently across multiple lines from these different wild-derived animals that are consistently differentially expressed. So Ron and I simply grabbed these gene lists, or the portion that you list here, and put them in the NEMO analytics system online, so we can actually look at them in other data sets. And so this is your Cas gene list, just those, whatever it is, 6 or 8 genes you had next to the heat map, painted on mouse egg cylinders. So 3 different mouse epiblasts, where the top of the plot is where they implant proximally, and the bottom is distal. The right side is posterior, where gastrulation is beginning, and the left is anterior, which will be the presumptive neurectoderm. So the genes being overexpressed by the cast line, here FOXA2, the couple GATAs, HEX, BMP7, Cerberus, are not only expressed on the posterior side of the animal where there's gastrulation, but and even quite biased toward the distal end.
Mhm.
In contrast, when we look at the PWK signature, which is again 4 or 6 genes, it's again on that posterior side, but proximally now. So this was a way we were trying to sort of connect your observations to a lot of thinking that we started doing when Silke's paper came together, of mapping human pluripotent variation onto the mammalian development map, and then doing the same thing with what you're getting into, these mouse lines that are outbred that have 1 million years of difference between them and are behaving differently, and mapping those differences at this early stage, right? Primed pluripotency is as close to the in vivo epiblast as we think we can get. So again, just taking your, what I think, in your paper, in your notions, is a baseline identity, a baseline observation of what these cell lines are doing, and mapping it back onto development to see what actually is the cast line doing versus the PKW versus the black 6. And this is
that's great. Yeah, no, I think we have these more refined subsets too from the single cell data that we've been working on, that I
wonderful.
think would be even better way of looking at that, because I think the concern with the bulk data is that you're sort of averaging over these maybe different substates,
couple
and
different
that
things happening. Yeah,
the frequency of those substates might be variable between the lines, but so that we have, I think, signatures for each of the substates now. And I think at the single gene level, we've looked a little bit on these what they call the corn cob plots. But at the single gene level, it's a little less clear than what you're seeing here with the signature. So that's really interesting.
Yeah, we'd love to get into those gene lists. And also, by the way, these the tools we're looking at are totally public. You can visualize any individual gene you would like, as well as any gene signature. I don't think we have time now, but absolutely, if any of you want to be able to do this, please reach out, because this is a page that has tens of different studies in mouse development. This is the Pijuan Sala data showing the same gene list now. This is the CAST gene list. Here's the corn plot, and here it is across every 6 hours of mouse development. This is Bertie Gutgens and John Marione's data, led by Pijuan Sala, way back in 2019. But again, painting these things on, here's that early, both primitive and definitive endoderm that this signature is showing up in, as well as later cardiac fates. Again, this is the CAST bias signature. So yeah.
Oh, that's great.
I'm happy to share these resources.
And the top is from the Naihajing. That's actually the corn. You're not regenerating corn cob plots. You're using his data.
That's exactly right. It is their data.
Okay.
Yep. Yeah. So this whole project, Nemo Analytics, basically tries to get gene level data from published studies, just so we can look at it all in one place. Yeah.
Awesome. No, that's great. Yeah, I think that there's more and more data coming out, and it's just hard. It can be hard to
absolutely
cross-reference everything and analyze the different, they have mouse and human differences too, and things like that. So,
that's one other thing we did, is that this CAST signature, this is now Silke's pluripotency data, where we had identified one of the lines, this is I4, that has very different differentiation properties than the other cell lines, and it totally overexpressed. It looks like a CAST line. This is 2, 4, and 6 days within pluripotency. It's ramping up this same signature, this FOXA2 and friends signature that no other lines are expressing, even if you put it in BMP. So it's something that's happening really specifically in pluripotency.
Oh, that's cool. Yeah. I think one of the main signatures that pops out from our single cell data is this sort of more epithelial versus more mesenchymal is not the right word, but like less epithelial. And that's sort of what Elisabetta Ferretti's paper was looking at, is this, they have a marker claudin 6, which is expressed in kind of more epithelial cells and is biased towards the anterior epiblast. But we do see this kind of co-regulated signature of genes that some of which are associated with more mesenchymal-like phenotypes. And I think that had been characterized also in Martin Pera's work, where they see some of his genes sort of overlap with this signature as well, where it might represent like dense clustered cells in the interior of colonies versus the exterior cells, which there's a lot of work about how the epithelial biology of like the colony of cells like causes the outer cells to behave differently than the inner cells. Because you know they're more tightly packed on the inside. The outer cells have basolateral BMP receptors that are available. They might have like beta-catenin differences that cause them to be more responsive to Wnt signaling. And there may be also just more stretched out, which again might have to changes in things like YAP, TAS activity. So I think, you know, there's kind of very overt different, if you look at a plate of stem cells, right, you can already see that they don't all look exactly the same, and kind of trying to figure out how that correlates with these molecular signatures and how, you know, one might strategize how to minimize or maximize those different subpopulations, you know, is I think a very fruitful area, because I think they do really impact the ability of the cells to differentiate into specific, you know, downstream lineages, depending on what kind of phenotypes they have in the pluripotent state, of course. That's great.
Cool. Yeah, I'll send a link around to some of these resources.
I have to leave. I'm
Awesome.
sorry.
Oh, no,
No
thank
worries.
you. Thank you for your questions.
Yeah, thank you very much.
Yeah, no, thanks so much for the invitation, Carlo. And
it was wonderful to discuss it with you. Thanks so much for coming.
Thanks so much. It was a lot of fun, guys.
Yeah, nice to meet you, pal.
See you, pal.
Yeah.
Bye.
Oh, see ya. And you're going to post this online
yeah, I
soonish,
try
or
to do
how?
it I try to do it as soon as I can save it and pop it up there.
Awesome. Great. Well, thanks so much. And
likewise,
I'd
Tommy.
follow up with you about this NEMO pipeline as well, because I think that would be very helpful for some of the things we're doing.
We're trying to make it easy to look at as much data as you want to. So, and I'd love to get your other gene lists in there. I can show you how to do this, or if, you know, we try to make it as simple as possible to look at as much data as possible.
Yeah, no, that's great. I'll probably follow up with you. The next 2 weeks are bad, but in the new year, I think we can catch up and find a time to talk about that more.
Great, Tommy. Thanks again,
All right.
man.
Yep. Thanks so much. Take it easy.
You too.